Hello, listeners. Yamina here. I'm very excited to have you join us for another Dr. GPCR podcast episode. If you haven't heard, we are officially a nonprofit 501c3 organization. Becoming a nonprofit only solidifies our mission to serve the GPCR community. Therefore, we're taking a short podcast break starting in July to come back with new episodes at the end of August, as well as an amazing set of events in the fall. If you are an ecosystem member, by now you know that every Thursday you get your weekly news or weekly GPCR news delivered directly in your inbox. If you're not an ecosystem member yet, join us today. Thanks to our fantastic team of volunteers of GPCR scientists, a GPCR article will also hit your inbox weekly on Tuesdays. Also, mark your calendars for not one, but two Dr. GPCR University classes with Dr. Terry Kanakin. The first one is titled Techniques for Effective Lead Optimization of Candidate Molecules, Principles of Pharmacology for Drug Discovery, which will start on Thursdays, September 5th at 10 a.m. EST and run for 4 consecutive weeks for 90 minutes. Then, on October 3rd, an advanced class will start about therapeutic translation of GPCR activity, advanced method for the optimization of candidate selection. This class will run for 5 consecutive weeks, also from 10 a.m. Eastern Time on Thursdays. For both courses, we will be offering 5 scholarships per course to GPCR scientists who live and work in developing countries. So stay tuned. Just a short week after the beginning of our second Dr. GPCR University class in the fall with Dr. Kanakin, on October 11th, we will be hosting our Dr. GPCR Symposium on GPCRs as therapeutic targets. During that day, we'll have talks from speakers of all levels, both from industry and academia, a poster session, as well as a panel discussion. We're also excited to be co-organizing our very first in-person adhesion GPCR workshop in collaboration with Dr. Anthony Boucard from the Adhesion GPCR Consortium. Mark your calendars for October 23 to 25 and join us in Mexico City for a fantastic meeting on all fascinating aspects of adhesion GPCRs and more. If you'd like to sponsor the Adhesion GPCR workshop, please email us at hello@drgpcr.com. Both emails, actually, we have now 2 emails, hello@drgpcr.com and hello@drgpcr.org. Both of these emails will be active for a foreseeable future to make sure that we don't miss your emails. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I'm delighted to have with me Dr. Nicole Perry-Hauser. We finally got together to talk. Today was a hectic day, but we're here. Hi, Nicole.
Hi, Yamina. Thank you so much for having me. I'm very honored to be included on this podcast.
Thank you. I'm excited to have you here today. And I forgot to ask before we hit record, do you like Nicole, being called Nicole, or Nikki?
I like to go by Nikki, but Nicole is on all my documents, so I'm fine with either.
Okay, so I'll stick to Nikki then, since that's your preference. And I've been calling you Nikki before, and for some reason, when we started, I just read your name like I didn't know it. All right, super. So let's start at the beginning. Would you please walk us through a short introduction about who you are and as a scientist?
Sure. So I am currently a postdoctoral research fellow in the lab of Dr. Jonathan Javich at Columbia University Medical Center. And I've been here, this will be my 3rd year in September. Currently, my research focuses around adhesion G protein coupled receptors, or AGPCRs. But before I came to GPCR, I was working with the arrestin proteins, and I did my graduate research at Vanderbilt University under the co-mentorship of Dr. Seva Gurevich and Dr. Tina Iverson. So it was a nice split between biochemistry and macromolecular X-ray crystallography.
Nice. I knew that you were a postdoc in Jonathan Javich's lab. I didn't realize that it was quote unquote just 3 years.
Yes,
I feel like I've known you for such a long time from last year when we met, when you guys were organizing the transatlantic meeting for early career researchers and stuff. Right. So it's a small GPCR world.
Yes, and a lot of time seems warped after the pandemic.
Yes.
I can't believe that I am also a third year. It feels like I've been here for a long time. It also has gone by so quickly.
Yeah, I think the pandemic just, put a lot of things into perspective.
I agree.
So you mentioned that you completed your graduate studies, your PhD at Vanderbilt University before going to do a PhD. Where were you, and how did you get into, science in general?
Sure. So I started at Wittenberg University for my undergraduate. Most people have never heard of that institution. It's a very small liberal arts school that was near my hometown in Ohio growing up. And so I went to Wittenberg. I double majored in chemistry and biology. We had very generic majors. We only had biochemistry, biology, and chemistry as our main science focuses. And it was there that I really got involved in research. So I've known since a young age that I'm very passionate about research, but I was also just passionate about learning in general. So I find a lot of topics extremely fascinating. But the first project I really started out with was in a entomology lab. So I worked with Dr. Jay Yoder, and we looked at adult female ticks and how they responded to tick excreta, in particular the component guanine. And that was a pretty disturbing research experience, I would have to say. But I really liked asking the scientific questions and getting to test it. So I did quite a few other research stints in my undergrad. I won't go through all of them, but because we were a small school, a lot of our focus was ecology based, and we were limited. We didn't have access to grad students or postdocs, masters, or really anything other than undergraduates. And it's since changed since I graduated. But so what I found most impactful was my junior year, I was accepted to an REU, which is a research experience undergraduate at the University of Tennessee in Knoxville. And it was there that I got to meet postdocs and grad students and master students and really see what it was to be involved in research. And that was my first genuine exposure to a research laboratory. And I loved it. I fell in love ever since then. So it really confirmed that I was making the right decision. So that's kind of how I got into science in very brief.
I love it, and I can hear it on your voice and the way you're explaining it, that you really, that was an eye-opening moment to you, and then you felt like you were in the right place.
Yes.
I'm intrigued. You mentioned doing different stints in different research labs at Wittenberg University. Anything that stands out? Anything that got you thinking about, okay, I don't necessarily like this, but I like the concept of doing science and research?
Yeah, so I would say I was pretty easy to please. There wasn't much that I didn't like doing. I tried my hand at organic synthesis in my OChem lab. I worked with zebrafish and developmental biology in another lab. As I mentioned, the tick lab was actually a microbiology group. I really found nothing tedious, and I think that's because we had limited access to these, big labs. And so any opportunity I had to do anything, whether it was a gel, a Western blot, or make this buffer, make the LB, I was just excited to do that and to have a place in that lab.
Awesome. It sounds, and the tick lab sounds very intriguing, because other than the fact that they're horrible to get off of your skin and then you can get Lyme disease, I think most people don't even think about ticks in general.
Yeah, he was he didn't have a lot of competition, let's say, in terms of other people working in this group. And for someone at a small institution, he was very he was publishing quite a bit. He was going to conferences. So it's still a great experience at these small schools. I think the advantage to small schools is you do get a lot of hands-on attention.
Yeah.
And you have to work with the materials because there's no one else there to work with that for you. And so you get a good approach of people directly mentoring you, and I think that really helped me when I moved to Vanderbilt, where it was completely the opposite. It was a little bit more hands off. You work with the post. It's more traditional in terms of what you're used to as a research environment. And because I had that strong foundation at a smaller school, it really helped me succeed there.
Yeah, it sounds like it gave you the the confidence, built out your confidence, and then you knew that you were capable of doing these things. You had the support you needed, at Wittenberg, and that allowed you to move on to the next level.
Yeah, and I would say that didn't come easily. I think my first year of grad school, I was very timid because I did come from a school that people didn't really know. And so it's intimidating, and you get that imposter syndrome that everyone likes to talk about. But after a while, you grow more comfortable and realize that your education is just as competitive as those at, more brand name, larger universities. And I felt like after the first year, I was able to catch up to my colleagues in terms of the research abilities and understanding the material.
I think, and it goes to the passion that you had at working in the lab, and knowing that, you're interested in this and this is what you want to do,
Mhm.
which is a motivator when you know which direction you want to go into.
Right, right.
So tell me a little bit, before going into for your undergrad, you mentioned chemistry and biology. As a child, what did you want to become?
I just love to learn. My mom has one of the copies of my composition notebook where I wrote down in fourth grade that I was going to solve all the environmental issues. I have never studied environmental issues since then, but I apparently wanted to solve that. So I would say my curiosity and scientific interests were almost always present, which was strange because my mother is a Spanish teacher and my dad worked in a printing company, and science wasn't really a big thing in our family. But I grew up in Plymouth, Massachusetts, and we were by the beach, and loved to play and observe the marine life, and just really was an investigative child. And I think that's what sparked in me a passion to want to continue to explore that.
Love it. I love it. And how did you get into studying GPCRs? So you went from Wittenberg University to forget I Vanderbilt. Yes,
Yeah.
that's the one I was looking for. And then what happened? You mentioned that you needed to adapt, going from a small group to a larger university.
So I would say in undergrad, I had no clear trajectory in terms of what I was interested in. And the reason I was so attracted to Vanderbilt is because they have an interdisciplinary graduate program. So you come in and you rotate through 4 labs in any of the 11 departments at the university. And so it's really open. My first rotation, I did microbiology. My second rotation, I did microRNA work. And then my third rotation, I ended up with Seva Gurovich, and we just got along so well and clicked. And I really enjoyed just being in his lab. And he was also working in the lab, which I thought was really cool, to work alongside your PI. And afterwards, I rotated with Tina, who actually just works down the hall, and she does a lot of the structural components. So and Seva and she had worked together successfully before, so I thought, why not do this co-mentorship again? And that's where I got focused on the Arrestins. So I really stumbled into the GPCR world. I never actively chose it. And ever since I started with the Arrestins, I was going to GPCR meetings, being involved with all those names. I always felt like the strange child at those meetings because at the point I was starting, I don't think Arrestins were as blown up as they are now. Now they're becoming sort of just as well known, maybe not colloquially, but around the community.
Yeah.
And so at the meetings, I was always fascinated by the GPCRs, but still, farther downstream. So I never really zeroed in on how GPCRs work in terms of all of their structural components until I entered the Javich lab at Columbia.
That's so interesting that you mentioned that at the time, Arrestins were just something that some people were studying, and now it's receptor, beta-arrestin, G protein.
Yep.
And there's other components that other people are studying, but whenever you think GPCR, you think beta-arrestin and G protein. So
yes,
I think with
time,
please.
Hopefully the GRKs are coming up in there too.
I think they're a good third or fourth in line when it got after, third beside the receptor.
Mm-hmm.
But I think there are other interesting subcellular, components, and some other intracellular components that are important for GPCR function that people are studying. I'm thinking of Joanne Trejo, and we're getting there. We're getting to that picture. So you mentioned that you kind of stumbled upon the field and you started out working on Arrestins. Do you remember the first time you heard about Arrestins, or GPCRs? And if yes, what was the situation and what caught your interest?
I think the first time, if I'm being truly honest, was in my senior year of undergraduate in our biochemistry textbook, where I think GPCRs have all of a page and a half of the whole book. And so I knew of them, in terms of studying for my final and knowing that, they couple to G proteins and they signal, and there's this thing called arrestin that's important for stopping. And that was pretty much it. But then, of course, Seva gave a lecture in my first year of grad school. And that's where I sort of, I just fell in love with Seva. We clicked very well personally. And so I wanted to go and see what his lab was like. And we got along really well there too. And so then I think because Seva was so passionate and excited about Arrestin and GPCRs as a topic, that I also fell in love with that as well. And then just going to my first conference or set of conferences, the community is so incredible. And I look forward to going to those conferences and seeing those people again. And now it feels more like a family than anything else.
Agreed. And hence, hence Dr. GPCR and everything that we've been doing and the ecosystem. And it's kind of one of the reasons why I started the podcast, but also which it developed into all the programs we have. And now we're going towards this virtual playground, is because to me too, it felt like a family. I always felt like at home, like, and building that virtual home for this family becomes very important to me, because we're all dispersed all around the world. We all do our thing, and then we get together from time to time in person at conferences, and then COVID comes along, and then we're all like, what are we doing? Can we please meet? So a nice way to help the community, I think, is really having this playground, because it saves time. You're looking for something, you know where to go.
Yeah, and I think the ecosystem has been great, and watching it evolve over these just few years that it's been present.
2.5.
Yeah, it's been really great. And I think that was also the push for the conference that Brian Bender, myself, and Andreas Brock and Buzzy Nisheva put together last year. But ours was, of course, more trainee focused. We wanted the early career people to really have a space to practice and present, and we wanted it to be free. A big part of it was we wanted it to be free and accessible so that they could attend. I think a lot of the GPCR meetings in general are expensive, especially if you have to go in person. It could be, $1,500 just to travel and take room and board. So having these opportunities for people to present, that they can go to their PI and say, I'm going to present and it's free, I think is an important.
Yeah, and I think you make a great point about, creating the first transatlantic meeting I think it was great. I love the fact that it was trainee focused. I love the way the program was put together. This year was also a success. I was on there on both days. And when we initially thought about in 2020 about putting together a virtual Dr. GPCR summit, it's funny because the thought came in August and we were like, let's do it in September. So we had a month to pull it together, and just how amazing the community is. We had 30 talks and it was pre-recorded, and all the emails coming and going, and we had talks from all around the world. We had high level people who were willing to give their time, and we also made it free the first year. Last year we tried to change the format, and it was also successful. And last year we had live talks and pre-recorded talks. And my big thing was not to separate PIs from early career researchers from trainees. And what I realized is that there is that separation, and I think it's a matter of pushing trainees, postdocs, masters, and PhD students to come forward and be able to present live on Zoom virtually after or before, big names.
Yeah.
And last year we had...
That's scary.
It is scary, which I understand, but that's why I'm pushing people. And We had 6 amazing talks last year at the summit. And now we're preparing the 3rd edition of the summit, which is going to be in October because of all the GPCR meetings in person popping up.
Right.
And this year we decided to make it free for everybody with people and ask people to become site members on the ecosystem. So you go online, you sign up, and you become a site member, you get approved. So we want to make sure that the ecosystem and people who are in the ecosystem are really GPCR people. And that's also free.
Right.
So it's a nice way to have that. And the novelty this year is that we're going to have a full day of trainee talks.
Oh, great.
Which I think I didn't want to have that separation, but I think the field is pulling towards having that, dedicated day, which is, I think, fine. We're going to have posters, we're going to have trainee talks, we're going to have workshops on how to analyze your data. And also, Nicole Smith had accepted to give a talk about, or actually a workshop on how to preserve data integrity and how to be organized in the lab based on her own experiences with having to retract papers because of a sequencing.
Right, I do remember that. Yes.
Thing. And, and I think it's important. And all of this, I'm saying all of this because this would not be possible without a phenomenal GPCR community.
Yes, I do think what was apparent to me when I was organizing the Transatlantic Symposium was just how much of an overwhelming response we had from people who were in the field for a long time, that they want to talk to the trainees, they want to be involved.
Yes.
And I find that super encouraging. I think it is intimidating at first to talk to any of them because they're the people whose papers you read and who you just look up to. And so it's bridging that first step is difficult. But then once the ice breaks, I think people feel more comfortable. And I think they did a really great job this year. With the panel discussion they put together, It just made it. And they asked trainees to submit questions before. And I think that really, really helped people who wanted to ask questions but maybe felt more nervous. And then it really did get people asking them in real time.
Yeah, I agree. I listened in on that one. I really liked the fact that, people were really interactive and there were more questions than time to answer them.
Right.
And yeah, it is a very good problem to have. And it's because we did that as well last year at the summit, and we had put in an hour and a half, and we had the same thing, which is amazing because people are interested in learning more about, how to develop yourself, how to go into the career that you want. But at the same time, higher level panelists were there giving their time and giving back. And I think it's a good field to be in. I think it's a wonderful field to be in. All right, let's get back a little bit to you. I think we kind of went away, but this was all based on your comment that how nice people are in the field. So you mentioned that you've worked on Arrestins, and then now you're working on adhesion GPCRs. The question is in the episode outline, and the question is typically, what is your favorite GPCR? But before you answer that, I put in here, it could be a receptor family, but it can be a G protein, it could be an arrestin, it can also be a phosphorylation motif, because I think it was Andrew Tobin or maybe Graham Milligan who said, well, actually, I do have a favorite phosphorylation motif. And I thought this was such, taking it into such a level of nerdiness, but at the same time, I love it. So any favorites in the family?
Well, I will have to say I worked with Arrestin 3. And I do use Seva's nomenclature, sorry. So beta arrestin 2 for those other listeners for a long time. And so it used to be my favorite, but now I'm I don't want to look at it for a few more years. I need some distance. And so I would say, because I've been working with actual GPCRs for a very limited time, I'm biased, of course, towards the ones that I'm working with. And those are the latrophilin receptors. So latrophilin 3 is probably where I put my most efforts, and that's more involved in neurotransmission and it's in the brain. But I look at all 3 of them, so 1, 2, and 3.
Mm-hmm. But I like how you mentioned that you're biased, and it
Yeah.
kind of goes to that. How well do you know beta arrestin 2 or arrestin 3? Would you be able to look at the sequence and be like, oh, this is what it is?
Yeah, Seva is he's been in the field for a long time and he still keeps a lot of his clones on regular paper. So I would say that I have become intimately familiar with a lot of the sequences. Um, we have since kind of brought him into the, digital age, but
That's
he
good.
still has those reliable constructs and he loves to sit down and do them by hand.
Wow. Well, that's really awesome, actually. So how did you get into the Javits lab, and how did you get into studying adhesion GPCRs, the lateral receptors, in your current lab?
Yeah, so I joined Jonathan's lab shortly following graduate school. I think I graduated in May and then went straight into postdoctoral work. And Jonathan laid out 3 projects for me and said, which one interests you the most? And basically, they were the 3 major topics that Jonathan looks at in his lab. And one was with the M-glue receptors. One was with single molecule work, which they've recently published. And the other was the adhesion GPCRs. And there weren't a lot of people in the lab working on it, just one other person. And I just thought they were so interesting because a lot of the adhesion receptors, particularly the latrophilins, don't have small molecule ligands. And so that makes them very difficult to work with and study because a lot of our GPCR assays are acute activation assays. And so that was an issue with the latrophilins. They have endogenous ligands, but they're mostly transmembrane domain proteins. So they have endogenous flirt and tenurins, but these are also very difficult to work with. So we, when I first entered the lab, we're looking at a way that we could acutely activate these receptors. And so that was one of the first papers that came out of my time with the Javits lab was the first author was Sina Mathiasen, and she's since become an assistant professor at Copenhagen University. But she used a thrombin cleavage site and kind of put it right in front of the CTF so that she could use thrombin to enzymatically activate the adhesion receptor. And it was really, really successful in terms of using it in our acute activation assays. And so I've been working on optimizing that so far in my postdoc.
I love it. And I think they're super interesting receptors because they have these super long extracellular,
Yes.
and terminal domains, and then you have the gain domain and then cleavage sites, and it can be it's just a messy thing to be working with, but I think that's the fun part of it.
Yes, and this year has been extremely exciting. As you probably know, there was a quartet of papers that were published in Nature, April, I believe, of this year, and it totaled up our adhesion GPCR structures to 8, as opposed to we had 1 that came out, I think, in 2021. And they were all truncated, so none of them had the N-terminal portion. They're all truncated to the CTF. But it's the first time that we've ever visualized where this cryptic agonist, the tethered agonist, interacts on the receptor. And it was just wildly conserved in the center of the receptor. And so that was really cool to see. And what was exciting is the adhesion GPCR workshop was the next month. So a lot of the authors were at that workshop, and it was fun to talk to them and discuss what they thought about the activation.
Yeah, I'm a big fan of the field and of adhesion GPCRs, and which reminds me, you mentioned, the structures that came out. So Ines Liebscher will be presenting at the summit this year. Excited to have her on. Anthony Boucard, who's also an adhesion GPCR person.
I met Anthony at that workshop, and I've never met a happier scientist.
Right? He's just amazing. He's just amazing. And he'll be also presenting at the summit and the next meeting in 2024 in Mexico.
Yes. Yeah. We're very excited because his lab is in Mexico. So it'll be great to kind of tour where he works.
Exactly. So the big secret, which I'm going to spill out right now, and I hope you won't mind, is that we're co-organizing. So I'm helping to organize that meeting.
Actually, I think he might have hinted that at the workshop. So it might already be spilt a little bit.
Yeah, he asked me before, and he said, "You know what? If they agree to have me do it, do you want to help out?" And I said, "Of course." I think it's just what a lovely place to do it. Plus, I think it's an amazing topic, and I think it's going to be really awesome. And the big debate was beach or city, beach or city. And I think we landed on beach. So
Oh,
we'll
great.
see.
Great.
We'll see what happens. I voted for beach, and I think Anthony agreed with me. So excited to hear, to know more. I think we're getting a meeting together next week to see.
Wonderful. That's great. I hope to see you there in 2 years.
We will be there. We will definitely be there. So that's going to be awesome. So you mentioned having three latrophilins. What do they do, and what's the difference between the three receptors?
So the differences, I don't know if we have huge delineations in terms of their differences. I focus primarily on latrophilin 3, so I'm not going to dance around knowing all of the intricacies of 1 and 2. But 3, we're mostly interested about it in the nervous system and how it works in synapse formation. The Sudoff lab has published a lot on synapse formation with the latrophilins for both 2 and 3 in the hippocampus. And I think it's been, I mean, I love reading their work. I think it's very impressive. We're, of course, more interested in the downstream signaling mechanisms and how these signaling mechanisms might be linked to the adhesion. So I think an unanswered question, or maybe some labs have addressed portions of it, but I don't know if we have a complete picture of how adhesion dictates downstream signaling, or if they're even working together. So it will be interesting, I think, as we develop tools, and we're working on tools in our lab as well, where we can truly probe those two things concurrently and say, okay, if we have a ligand interaction, does that initiate intracellular signaling, or does some sort of intracellular signaling inform on how these receptors interact with others? And does that change synapse development?
I love it. And that's a very important and interesting question, which is, I think it crosses or gets out of the GPCR field. I can think of integrins. They're also involved in adhesion. And the question is, what happens? Is the mechanotransduction or the mechanosensing part of the integrins important to signaling? And how is that connected? Or is that two different functions that are independent of each other?
Right. And I think the mechanosensation is a huge topic right now. I think a lot of labs are trying to do sophisticated experiments where they can remove the NTF and look for signaling downstream. I know at the workshop that was a huge topic where people want to know, do you need to pull off the NTF to get signaling, or can it stay on there? And I think Enos actually just published a nice kind of piece where they commented on this, talking about how some adhesion receptors don't have that autoproteolytic cleavage. And if they can't cleave, does that mean they can't signal? And I think there is some biochemical work to show that the uncleaved receptor can still signal. And so that's interesting. And I think that brings us to the big problem that these structural studies have presented, which is that the tethered agonist forms a tight alpha helix when it interacts with the receptor. But when it's in the full length receptor, it's a beta strand in the gain domain. And so people, they want to know, how can it go from being an energetically favorable beta strand to pulling itself out and becoming an alpha helix if the receptor doesn't cleave? And so it's hard to imagine that. It's going to be very interesting to see how people explain that. I think Enos in her commentary did a good job, and it's going to take experimental evidence to really refute or support it. But I think it's a really compelling thing that's going to be the next topic that's hot for our field.
Well, 2024.
Yeah, maybe 2024, we'll know.
I think it's what I do very much like about adhesion GPCRs, and I think that's the difficulty again with it. It's the fact that they can be huge and bulky, and then they can do all of these things, and which makes you fantasize about, okay, what can it do? Why is it the way it is? What does it control? And how can we modulate it in disease settings? But I think the difficulty is the fact that they're very difficult to work with, and we don't have enough tools to really dissect out and get those answers.
Right. Yes. And there's domains in certain adhesions that we don't even know what their functions are. So it'll be very interesting. Like we know in the latrophilins, we have certain domains that are responsible for interacting with the endogenous ligands, such as the tenurin and the flirts. But there's a horm domain as well, and there's a pauline or a serine-rich repeat area. And what is the purpose of that? I don't know. And obviously, just basic things are difficult with these receptors. Cloning them is hard because they're more than 10 kilobases. And that's just my receptor. I don't know how big the very large adhesion GPCR is.
I think they go from 600 to 9,000 amino acids or something like that, depending on which one you're looking at. So when I think about the quote unquote poor little chemokine receptors I worked on, which were super easy to activate and express and clone and you name it, this is a whole new level of difficulty.
Yes, and you would think that it would be really amenable to cryo-EM, but I think with these studies that the majority were cryo-EM, none of them could see the N-terminal fragment. And I think that just talks about how difficult it's going to be to position that. And it'll be interesting to see what kind of tools they design in order to be able to, solidify it to visualize it.
Yeah. So speaking of tools, and it's funny because I did ask Anthony this, if he had any, if he had a magic wand and he can create a tool for him to, answer any adhesion GPCR related question, what would that be? And his answer was, become teeny tiny and go down into the cell and watch the receptor work, which I thought was very, I actually like that answer. And you mentioned, the paper where you guys have inserted that thrombin cleavage site in order to be able to measure receptor function. If you had a magic wand, what would you need? Like, what would you use it for?
I want something really basic. I want a stable cell line.
Okay.
Very doable. I've been trying to get a stable cell line of my receptor for a while, and for some reason it's thwarting me. I should be able to figure that out. That's the one thing I want so that I don't have to redo all the transfections. But I would also like a small molecule ligand, that I want an agonist, an antagonist. I want the complete set so I can do these pharmacology assays. That would be wonderful. And, I think a lot of groups have been trying to do high throughput screening for these receptors, and it's just extremely challenging because the readouts are hard. And it's hard to keep them consistent, which is another reason for a stable cell line.
Yeah. And the toolbox, you'll have your little toolbox, you have the vial of stable cell lines, you have your vials of agonists, antagonists, you name it, and then it's just a mix and read, and there you go.
Yeah, exactly. But, I think Anthony's answer is probably fair and would, get more solutions than mine. So maybe we should do his.
Maybe we should, I think there was a cartoon. I think it was called The Magic School Bus.
Yes, Mr. Frizzle.
Exactly. So maybe we should have a magic adhesion GPCR school bus, and we can go explore adhesion GPCRs or any GPCR for that matter. All right, super. We've talked a little bit about how amazing the field is and how nice and how giving the people are in the field. And I think, based on what you were saying, that you had a really great mentor in Seva at Vanderbilt, and you're really enjoying your time as a postdoc in the Javits lab. Any other advice for junior scientists, whether it's about how to pick their next step, how to, mentally go through a PhD and a postdoc, anything?
Right. I have my 3 things that I wrote down before, but something that's coming to mind is the fail to plan to fail, which I think is fair, but also unfair, because there's so many times that I've planned in science to do certain things and they just don't work. I interviewed with probably 7 postdoctoral mentors, and I chose Jonathan, and I did not interview with him. So, sometimes it's just you can't plan, and I love to plan, so that always is nagging at me. But I think for me, being successful in science, it's so important to stay positive. It's so easy to focus on the negative aspects of our job and the difficulties and, the inadequate pay. And those things are important, and I think that they should change, but it's not beneficial to me to focus on those in the here and now. And so I think when things are difficult and there's pitfalls and setbacks, it's the people I surround myself with who constantly encourage me and pull me back up. The main person of which is my husband, but also the people in lab who can sympathize with difficult things. That's super important to have people immediately in your community that you can reach out to for support.
And all of us listening, and me as well, because I feel like and you made a couple of great points. One about staying positive, because things never, or most of the time, don't work out the way you plan them. And sometimes you don't plan and oops, things start working and you don't know, you can't repeat it and you have no idea how that even happened. But also the fact that you mentioned, something important, and I don't think we've ever talked about this on the podcast, is the inadequate pay for postdocs and PhDs. And I think that needs to change. But
I would not complain to have some more funding.
Of course. And I think that's important because you are doing such a high level work and you should be fairly compensated to do that. And the potential explanation would be, oh, you're in training, so you shouldn't be expected to. But I don't think it's true. I don't think it's fair. That being said, I think knowing that you're not compensated to the fullest extent of your abilities shouldn't be something that prevents you from doing a PhD or prevents you from doing a postdoc. I think it should motivate you to stay focused, stay positive, and get out. In the sense that you move on. You determine what your goal is, you take what you need to learn there, you develop the skills you need, and then you move on. Maybe that's why it was developed this way. I don't know. I'm just speculating.
Although I do think we have to recognize that, there are people who can't just look by it, who maybe have families to support, and that does inhibit them from getting involved.
Yeah.
And so I would love to see that change so that we can have, more equality and more diversity present in our workforce, because I do think a lot of good people are deterred by that. They go to great places still, like a lot of my grad friends who, they couldn't afford to do the postdoc and would go to industry and do incredibly well and get the compensation and benefits that they need. But it's unfortunate for us because then we lose them as, an academic.
No, I agree with you. And I think, I've went through it with a family and it was not easy. And I think it was very shocking when, I left from a postdoc to an industry job doing pretty much the same thing at a different pace and doubling my pay. And I was like, wait a minute, all of these years I was worth this. And sometimes people feel like, you're being undervalued in academia. I don't know how to change the system. Maybe we should record a different podcast episode around how to change the system, and you're or maybe we should have a panel discussion about it. And I think it's unfair. And sometimes
it's a hard issue for a lot of reasons, because I can only really speak to the US experience that I've had. But the NIH funding is limited too. And so even though some PIs may want to pay more, they might not have the ability to pay more.
Yeah.
And it's just becoming, it's not as straightforward as let me just increase your money. It has to come from somewhere.
Yeah.
But I do hope, and I hope that when I'm a PI, I remember, how difficult it is to situate yourself in some of these cities that are most attractive for postdoctoral candidates. Like I think of the San Francisco area, I'm in New York City, but Boston, these places are not inexpensive. It takes a lot, and you do sacrifice to be there. And I think, like you're saying, for some people, the sacrifice feels like motivating to get in, get out, and do what you need to do. But there's always going to be people who can't do that, and it's not because of lack of passion.
Yes. I agreed. And it's a difficult choice to be made. And I think you made a great point, as I don't think it's the PIs who don't want to pay students and postdocs more. It's the way the system is. And for example, I had a pretty long podcast episode with Stuart Mosley, and we were trying to figure out how and why is success measured the way it is measured in academia. and what can we do, or how can we change it in order for it to be a little bit more democratic, I want to say, and then not lose all that talent. And I think this goes back to the same funding question. It's the system, but we still have to talk about it.
Yeah, and hopefully change, make little
Yes,
changes as we go.
exactly.
But that kind of brings me back to the stay positive thing. It is really easy to focus on all these hardships, and I completely sympathize with people who I see, exiting academics, and we seem to be in a period of mass exodus right now from academics. But there are such wonderful positive things to fight for, and that's a reason I want to be a PI. I want to be there to help facilitate some of that change. And it's not going to be easy, and times it's not going to be fun. And there are days when I just think, man, I should jump ship. But I try to make those days less than the positive days. And I think what helps me is mentoring people in the lab, and, especially young people. Not that I'm ancient.
Yeah.
But,
25 plus.
Yeah. Yes, thank you. But like seeing these people who are getting introduced to science for the first time, and the excitement they get with their first result, and that's what I want for people when they come in the lab. I think the longer that people are in the lab, sometimes the more down they get about the system that we're working with and the flaws that it has. And seeing people come to science for the first time is what keeps me motivated to make those changes.
I love the point, the couple of points that you made. I think I've always enjoyed having people visit the lab. I had, when I was doing my PhD, regularly had high school students come in the lab. There's two boys, two young teenagers, 14, 15, who spend the whole day purifying DNA from E. coli. And I don't know if I cannot remember their names. I have no idea. All I remember is that they left with a sample of the DNA they purified, and we had quantified it, and they were the happiest kids ever.
Yes,
And
that's what I do in the morning and hate doing, right?
Exactly. Well, they were working, they made my DNA, which I was like, okay, there you go. It was great experience for you. I don't have to do it. But I think that's important. The other point that you made, about kind of jumping ship and really focusing on what's the positive. And I think what's important is to figure out what is it that you want to do, and figure out a path to get there. And I think right now, with the current environment compared to 10 years ago, it's much easier to look at your options to potentially go to industry. But for a long time, at least in my mind, it was like, if you're leaving academia, you go to industry, there's no way back. And I don't think that's the case anymore.
No, I would agree. I think even when I was at Vanderbilt, we had several people who had returned
Yes.
from industry. So we had Jeff Kahn,
Yes.
we had Dave Weaver. So we had some really positive, successful role models who showed us that there are ways to marry, the industry feel and the academic desire to kind of probe a little bit more deeper.
Yes. Yes. No, I think it's important. And I think the other change that I'm seeing is, for a long time, at least, again, in my time or in my head, and I've been 25 plus for a while now, is that people would say, oh, it's academia or industry, and the industry or the private sector is the dark side. And I don't think it's true. I think it's the other side of the same coin. And again, going back to the ecosystem and to the fact of how fantastic the GPCR field is, I think if our goal as a field is to improve human health by doing everything that we're doing, we will benefit from coming together into the same playground with biotech, pharma, suppliers, and talk to each other. And the goal of the ecosystem, the way I like to explain it is, imagine this beach, you have all this sand, and each grain of sand is somehow like dispersed on the beach, and we have GPCR folks and GPCR companies, and the goal of the ecosystem is to really kind of bring everybody together who works on GPCRs, who has an interest in improving human health through targeting GPCRs. And I keep saying GPCRs, but that's not exactly true. And it's GPCRs in general, and it involves beta arrestins and G proteins and anything that's related to the field. Sometimes people tell me, it's Dr. GPCR podcast, so I'm working on G proteins. Can I come on the podcast? And yes, the answer is yes. It would have sounded weird to call it Dr. GPCR, beta arrestin, and G proteins.
Yes. And I think getting all these hats in one room is very important. We all think very differently. I think a lot of academics tend to be in the nitty gritty, and because industry has to sell a product eventually, they tend to be more big picture. And so having those together is super beneficial. I also think the smaller biotechs that are cropping up really do seem like little labs to me. So, we've kind of got a melding of all the worlds together, which is fun to see and live through and explore all those possibilities.
I think and I think you make a great point. I think the smaller biotechs are like these, rocket fueled, labs where, yes, the goal is at the end of the day to make money long term, but it is very data driven. It is very science based, and I want to say the main differences between an academic lab and a biotech lab, whatever topic they're focusing on, is that in biotech you have tighter deadlines, so the speed is much faster. You have well-defined deliverables, or most of the time well-defined deliverables, and funding is different as well. Because the goal is not to teach, the goal is not to show that you can do experiment X, but the goal is to get data fast so that you can succeed and or fail fast as well.
Right.
But at
I
the end
wish sometimes that we had better deadlines. Then I'm a deadline maintainer, and so when I get collaborations with people who like to, extend it up to the last minute, or we can turn it in a little late, it's fine. I hate that. Drives me nuts.
I feel you. I don't, pushing stuff down the line just because we can push it down the line, it's something that I don't particularly appreciate. We talked about it. This is it. We've planned it out. Sometimes when things happen and, some data takes you into a different direction, it's totally understandable. But when it's just out of thin air saying, oh, we can do it later, that just drives me nuts as well. So
we're definitely type A people.
Yes.
That's okay.
Yes. Organized, list-based, deadline-based, and, there's no going around things. Any other advice for junior scientists?
I think something that I was told that I think is still important is to practice writing consistently and all the time and every day. And I've always been grateful. My other mentor, Tina Iverson, was a big proponent of having us write our own everything. So I think a lot of other PIs tend to be more hands-on. Maybe they'll help really drive your first few papers. And Tina was the opposite. She would have you really write everything. And she gave phenomenal feedback for you to kind of readdress and rewrite and rethink. And I'm so grateful to her that she did that. I did not like her at the time because it's a pain in the butt to have to do all of that foundational work yourself, or yourself with pushes in the right direction. But now I'm so grateful to her because when I go to write my grants, or go to write a paper, or go to write answers for a podcast, I feel like it comes a little bit easier. And you learn your scientist writing style, because I think it's different than other creative writing styles or even colloquial writing styles. And so I think any opportunities that you're given as a student to practice, whether this be grant applications you think you have no chance of getting, or op-eds, which are opinion pieces, or, just sitting down for a few hours a day to write down your methods, I think that's important. And it's a skill that can only improve with practice.
I love it. And I think also capturing what you've done in the lab, in a lab notebook on a regular basis, And now 20 days later, trying to scratch your head as to, okay, what was the buffer that I used and what's the catalog number and what's the lot number of things. But I think at least I had always a lot of trouble with capturing this information on time because I was always pushed to experiments. And working with live cells, it's just, you're at the mercy of your hex cells or whatever cells you're working.
And it's you have to be so, you have to really have intention every time. And if you are I think as grad students, when you're starting out, it's just like you're thrust into this and you don't really have a good example in a lot of cases of how to organize yourself, of how to organize your data online. And so you're trying to do all these things at once. So it's natural that you don't have time to sit down and write it all out. But now I think that I've become more senior. I'm very intentional about writing everything. And I like to keep an electronic notebook just so I can search through things and have a complete documentation. I'll write things by hand as I go, but I always update the electronic notebook at the end of every day. And I think that's a lot of work. And that's why a lot of people don't do it. But it has made such a world of difference when I'm going back and really need to know something because it's already there and I've already thought about it.
I think you made an amazing point about the electronic notebook. Back in the day, it did not exist. But nowadays, there's free tools out there that allow you to do so many things. And I think it's amazing that they're out there. You can use them and it's searchable.
Yes.
I think the searchable keyword, right? You don't have to be flipping through 10 notebooks and then you're missing notebook 5 and you know that the information is on page 102 on that and you know where it is because you can kind of remember which side of the page it was, but you cannot find the notebook. And now it's searchable. Or you can even ask Siri or Alexa.
Yeah, and I don't know. There's so many different flavors of these notebooks. So I think a lot of people can find one that works for them. And even if you can't use, maybe it's too expensive. I know some of these cost money. Some schools have subscriptions, so they're free. But even if you can't, you can make your own little digital kind of electronic notebook the way you want. Although I would say back yourself up to an external hard drive. Always have backup.
Yes. What, you mentioned electronic notebooks. Which one is your favorite? How many have you tried? Pros and cons, maybe people listening might benefit from it.
Sure. So I've only used Columbia's Lab Archives is what they have, and I like it. But I like to make my own version. We have a free subscription to OneDrive. So I do a lot on just self-organizing on there and then backing up to an external. That way it can be on any of my computers at all times. But I did enjoy Lab Archives for the little time that I used it.
Yeah, I've used Benchling, which is...
I've heard good things about that.
I think the ELN, so the electronic notebook, is free. The registries is out of reach for our academic labs, I want to say, because those are really expensive. But I think it's just amazing because it's searchable. You can share it. It's online. So, and it's backed up on their servers, or if you're in a company, then it's backed up on the company server, and then it's safe and secure. And it can even be, and it can be connected to instruments. So whenever, you finish reading whatever you're reading on your plate reader, just drag and drop and it's already in there.
That's super nice. Yeah.
Which I think it really helps with the data integrity. And again, the searchable, I think, is just such an exciting word when you're looking for something and you can just search for it.
And I will say, we also use, I'm recently, so I told you I started in biochemistry and X-ray crystallography. Now I work with mice, and it has been a wild ride. I never thought I would work with mice because I've always been afraid of blood. I was like the person who fainted in every single anatomy class. But I've since been able to overcome that. But the reason I say this is because we also use transgenetics as our online kind of mouse colony management.
Okay.
And that has been an incredible tool if we're talking about management softwares, because it tracks all your mice, it tracks your litters, it tracks your breeders. It's just been really nice. And you can share, genotyping information with your lab mates just in a click. And I think that was such a phenomenal tool.
Yeah. I have never heard of it. I have worked with mice. I did not like to work with mice. I think the most difficult thing was sacrificing the mice.
Mm-hmm.
And, you do it. I used to do it 2 by 2, and the first 2 was fine, but then the next 2 would feel it. All right. It was just, oh my God, it was the worst thing ever. I'm so glad. I'm the kind of person who had trouble throwing out hex cells because I had too many cells.
Oh no.
So like from being like, sorry, got to throw you out. It's Friday night. I only need 2 mils of you. To sacrificing mice, that was such a big leap for me.
Yeah, I actually, that makes me think, when I was mentoring someone, we were making LB media, and we were done with the media for the day. So we were cleaning it out, and we added bleach to kill the cells. And I told him, if you lean in closely, you can hear them screaming. And he believed me. And I felt so guilty. I was like, no. You're really
kidding.
Okay. But that's what made me think of feeling bad for your hex cells. That's a new one.
Yeah. I felt bad throwing them out, but whenever I was doing an experiment, it was important, and I would finish transfecting. I put them in the incubator, and I'd be like, "You guys do what you need to do. I'll be back tomorrow." And I don't know. I think I had this interesting relationship with my hex cells because everything depended on whether the cells would cooperate.
Yes. Yes, I did a little bit of work with primary neurons, and while I'm in Jonathan's lab, I've been doing that. And I thought hex cells could be finicky, but man, those primary cultures are very picky.
Wow.
So I feel you there.
Yeah, I've worked with different cells and cell lines and stuff, and I don't know. I always liked a challenge of bringing back cells from the dead, where you thaw the thing and there's like maybe 10 cells that survived, and then you put it in the 96 well plate, and then you wait a little bit, and then you transfer it into the 12 well, and the 24, then 12, and then 6 well, and then at some point it's going to pick up. But I think it's just I don't know, it was just a fulfilling moment when you brought them back.
Yeah.
Yes. they're back. They're back, and they're important. All right, top 3 aha moments that you had as a scientist.
I left these blank because I just don't know. I think they haven't happened yet. that was my answer to them.
I think based on what you were telling me, the fact that the aha moment was changing from a small university to a larger institution, where you kind of knew that you still liked what you did, but you might have felt a little bit, not at ease coming from a small environment, but yet you persevered. I think that must have been an aha moment based on what I've heard, is that you kind of overcame that fear, and then you developed confidence in your knowledge.
I hope I've developed some confidence. Is very hard in science, I think, because we are constantly humbled by the experiments that fail.
Yes. Yes. And I think, we're also taught to, at least in academia, to formulate things, to kind of protect, if just in case it didn't work like that, the data indicate, and we speculate and things like that. And it's never, I feel like when I used to write, it was never definitive. Which is fine, but I think that doesn't help when it comes to developing confidence in your data or in yourself.
Yes, and
because
I think we as scientists, one thing, when I was in grad school in our journal club, I remember that the professor that was kind of mediating our journal club for our class stopped us one day because we were reviewing a journal article and we were being particularly critical. And it's because we're taught to be critical, right? We're taught to look for what's wrong. And the professor stopped us and said, let's say some positive things first about this study and point out what the authors did really well. Because I think we do forget that a lot, and that has a lot to do with draining our personal confidence. Because we're taught to look for what's wrong.
Yes.
But and that goes back to my first point of you have to focus on some positive things too, because for a study where one thing goes awry, there might be 10 things that are meaningful and useful. And it, in the way that we publish science now, we have to publish a story. And I think sometimes it would be nice if we could just get our data out and not have, I mean, it's important to give background and perspective of why the study is important, but sometimes the data are negative and that doesn't mean nothing. And it is still useful. So I'm still trying to think of creative ways to publish negative data.
So we used to joke when I was in during my PhD with a friend of mine who's not in the GPCR field, is that we should establish the Journal of Negative Results. Because I think, well, again, and I'm bringing it back because that's also one of the reasons why the ecosystem is happening, is that so within the ecosystem you can join, become a site member, you can have a subscription to do it, and then it gives you access to all things GPCR and Dr. GPCR and everything in between, and then you get to talk to people, but at the same time you can create a course, or you can participate in a forum, you can start an adhesion GPCR, a latrophilin 3 forum, or a latrophilin 3 group, and this is potentially a place where people can put up their negative data. Because it's important to know what doesn't work, I think.
Yeah, and I mean that it would be great. I'd like to see this for all science. And I don't know if like a bioarchive is the answer where you can put data out there.
Yeah.
But I know people have strong opinions, yay and nay, for bioarchive. But I think it could be a very useful way to put data that might not have a home place where a student can still reference it in their CV or their resume.
Yeah. Well, we have to create it. We have to create the Journal of GPCR negative results for GPCR research. I think it would be super useful because, you're gonna save people's time, people's money, funding, knowing that, being very transparent. This is what we've used. This is the data. It didn't work. This is why we think it didn't work. And then, nobody will, potentially people are going to be like, I'm not going to repeat that. Or someone is going to read it and going to contact you and say, hey, have you tried this?
Yeah, right. And help you troubleshoot.
And exactly. And I think that's very important. And having a place where you can deposit this information and a place where you can talk about it with your peers and the community at large might be super beneficial. So let me think about it. I'll think about it and try to come up with something.
Do the synthesis. I'll just create the problems and you can solve them.
Perfect. And we're gonna give it to the GPCR field to solve that. All right. What else? What other aha moments?
Jeez.
Anything, I think the thrombin cleavage site idea.
And that wasn't my aha. That was definitely all credit to the first author of that study. And I just think now we've kind of improved about on that site. And I'm not going to share it yet because we have yet to publish it. But it was just such a remarkable idea that she and Jonathan kind of came up with. And I think really impressive way to get around the fact that we don't have a way to acutely activate these. And people before that were, they would remove the tethered agonist and then add it as a peptide. I think that was good as well, but peptides can be really hard to work with. And so having this way, I mean, I've used this construct design in a number of acute assays, and it's just so reproducible. And I would say, at least for our receptor, because I haven't used it ubiquitously across all the adhesion GPCRs, but it's just made working with them and probing their G protein coupling and hopefully one day their arrestin interactions possible.
It's an elegant system. I think that's the word that comes to mind. It's really elegant. I like it when people get very creative and they solve a major issue like this with something really simple. And you're like, why didn't everybody think about it 10 years ago?
Yes. And I will say, joining Jonathan's lab, Jonathan is he thinks of so many controls that I've never thought of that are he's really made me expand what I think of to be a good experiment. I used to say, okay, this is the positive, this is the clear negative, now we do our tests. And Jonathan will just think of all of these elegant additional negatives or positives that really should be included. And so I'm very grateful for him that he's made me kind of expand that mindset when I'm looking at my experimental design. But he's one of those people who really gets down in those nitty-gritty details, like you're saying, and really focuses on things that maybe after you're like, wow, this should have been really obvious, that weren't. And it's important that we have people like that who want to get down in the weeds, because they end up making these wonderful experimental designs and just coming up with these conclusions that would otherwise not have been reached.
Agreed. What's next for you? This is my last question. So you mentioned at the beginning, it's been already, quote unquote, already 3 years of your postdoc. What's next?
My ambition is to be a PI, and I'm going to ride that train till it crashes, I think. So I've applied for several grants, and, I don't think me becoming a PI is dependent on the success of those grants, but they would certainly help. So if you're reviewing, pay attention.
Yes.
But I probably will try to go in the job market within the next 2 years, I would say, and just start looking around. But if you ask me place, I have no idea. I don't even know if I would be bound to the US. There are certainly a lot of places, I think, particularly in Europe, that I find fascinating and exciting places to just pursue GPCR research. There's so many great hubs over there. And right now it's just me and my husband. So we're relatively free to kind of relocate and establish ourselves. So it's wide open, truly. I think I will continue to be focused on adhesion GPCRs. I'm trying to develop in Jonathan's lab kind of a slice of the science for myself. And Jonathan has been very supportive of, helping me move in that direction. And so just with a little bit more luck and some positivity, I'm hoping that I can achieve that.
Awesome. Well, I wish you all the best, and I cannot wait to have you back on the podcast, and so that you can tell us where you land. Obviously, it's an open invitation, and if you want to come back and talk about difficulties and funding and how we could revisit or at least talk about, funding scientists and academia better, in order to preserve it, because it's an important part of science, you're always welcome, Nikki, to come on board.
Absolutely. I don't know if I have any of the answers, but it's certainly something I can talk about.
Amazing. Well, with this, thank you, Nikki, for your time. I really enjoyed talking to you.
Thank you.
Don't go anywhere. I need a favor. Please share the Dr. GPCR University program with your friends and colleagues, especially if they live and work in a developing country. Please subscribe to our YouTube channel. Many of you come back to watch our videos, but aren't subscribed. Having more subscribers will help us get you more content. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest and our Dr. GPCR team, Attila, Ines, Montserrat, Ivana, Cam, John, Yatsu, Andreina, and Balint. If you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. And don't forget to subscribe to our monthly newsletter on our website to get all the news about all the work that we've been doing and we are doing at Dr. GPCR directly delivered into your inbox. Another great way to support us is to share your favorite Dr. GPCR programs with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com or hello@drgpcr.org. As I mentioned, both emails will be active for a foreseeable future to make sure that we don't miss your email. Until next time, stay safe.